Hello, it's a pleasure to talk to you at the SIU PPTC Northwell Pelvic Health Consortium, ICA Patient Consortium. My name is Joel Stern, and I'm an associate professor of neurology, surgery, and molecular medicine at the Zucker School of Medicine. And I will talk to you today about cytokines and exosomes in IC-BPS. And this is the current research that we're currently investigating. We want to understand what kinds of inflammatory signals play a role in the development of IC-BPS. And we want to find out if any chemical signals can be used to diagnose or even monitor ICS-BPS progression. This is a microscopic image of cells interacting in the lymph node. We have lymph nodes throughout our body, and the cells in green are T cells, and the cells in red represent our dendritic cells. And you can see how the cells are spending time interacting with each other. They're essentially talking to each other, and they're moving around, and there are multiple interactions that are taking place. And you have to think about when you have an infection or an inflammatory response, that your lymph nodes will swell. And this is basically what's happening. It's you're getting an inflammatory response due to the infection. And these cells will then recruit each other to the site of inflammation, which was the previous talk. And now we're going to take a closer look at the mechanisms of interaction in the next slide. So this is showing essentially a model of an antigen-presenting cell. In this case, an antigen-presenting cell is either a dendritic cell or a B cell, which we learned about in the previous talk. And here we can see that these antigen-presenting cells, or dendritic cells or B cells, present antigen to T cells. It's like they're talking to T cells. So essentially, from the last video that we saw, the reason why you have so many interactions is that the cells are trying to regulate one another. And this is done so that one cell is not overactivated and that each signal represents a specific circumstance. For example, when you try to start a car and you're successful, you have to turn the key, you have to put your foot on the brake, and then you have to put your seatbelt on. These are all safety checkpoints. Well, your immune system has a set of safety checkpoints so that you're not going to activate your T cells, and your T cells won't randomly destroy anything else around the surrounding area. You want to make sure that all of these are specific interactions. There are specific interactions between all of the cells that are involved. It's like when you put the car into drive or reverse, that's essentially like the cells secreting the cytokines to tell the cells where to go. So that's why we need other signals. This is, for instance, there are 3 signals that are required to activate a T cell. This one is, regardless of whether you understand whether this is, it's called an MHC molecule, which is essentially just a receptor. And this is a receptor on T cells. And this is an antigen. So the receptors have to engage one another through this complex. And then there'll be another set of receptors that will engage. This is just called signal 2. And then we'll get the cytokines or chemokines, and these are the chemical signals that will tell the cell where to go. It will give the cell the instructions of where to go. So essentially, the immune cells use these cytokines to communicate, and the cytokines will affect other cell types. And you'll get activation of the immune cells by cytokines inducing these symptoms that arise during the process of fighting an infection, such as when you have a fever or inflammation or the sensation of pain. Inflammation and these cells are always involved in that process. The study that we conducted consisted of 140 patients with IC-BPS that generously donated their samples for our study. And these samples consisted of urine, blood, immune cells from the blood, DNA isolation, and we were able to look at the cytokines and exosomes that are involved in cell communication that I'll show you right now, the data. So what we found was we looked for specific cytokines. These are IL-6 and TNF-alpha. These are just specific cytokines that are secreted by cells. And we looked at blood, and we looked at the concentrations on the left panel are concentrations, and below we're comparing patients with different IC-BPS, whether they had Hunner's lesions, which is identified as HL, and no Hunner's lesions that were present. These are both patients had interstitial cystitis, bladder pain syndrome. The UC is the control, the unaffected control population. So you always want to compare controls to patients that have other symptoms of the condition. And then we looked at both IL-6 and TNF-alpha, and we found significantly higher levels of IL-6 in patients who presented with Hunner's lesions, as well as increased amounts of TNF-alpha. So both of these cytokines were significantly increased in the urine of the patients. Next, we looked at in the urine, we wanted to see the presence of another way in which cells can communicate. These are called exosomes. And they have good and bad consequences, but essentially they're used for cells to communicate with each other. They consist of lipids, DNA, RNA, and they're found in the blood, urine, saliva, and brain tissue. And what we did is we isolated these exosomes from the urine of patients with IC BPS. And this is just a protein ladder. This is a blank. And we found the presence of unique bands, unique exosomes that were in the urine of patients that had significant course of disease. And they had more than several Hunner's lesions present in the bladder. And in patients that were in remission, you see a much lower amount of these bands, the presence of exosomes. So we concluded that exosomes are present in the urine during different stages of IC-BPS, and we're trying to understand the contribution of the immune system to IC-BPS. So we're approaching the challenges from multiple angles and using different samples in order to address these questions that we have. And we learned that the exosome analysis we may be able to detect changes in the bladder during IC-BPS progression. And we don't know if these changes are a cause or a consequence, sort of like the chicken or the egg, but this research does require more studies and more analysis. So I'd like to thank Dr. Rob Moldwin from the Zucker School of Medicine, who was influential in all of these studies. Our group from the Feinstein Institutes of Medical Research, Vishan Nursi, William de Gavia, and my postdoc, Micheline Laurent, from the Northwell, Mike Finaro, Oksana Yaksi, Sonia Balani, and Suhel Najjar from Rockefeller, Inga Tabansky, and Darren Keskin from the Broad Harvard Medical School. But most importantly, I'd like to thank you, the patients that participated in all of our studies, and without your participation, we wouldn't have been able to make these discoveries, and we wouldn't be able to continue to contribute to the field of IC-BPS. So I thank you for your help. And if you have any questions, my contact is provided. Thank you for listening, and have a wonderful day.